So we made these two particles. Actually, we tried to not make them too perfect because we wanted to look at differences. So we have a slightly lower grafting density of these polymers than we can optimally achieve, for example, to make the linear polymer particles good. But if we make them all the same molecular weight, same grafting density, we observe the following in terms of colloidal stability. Big coarse, 20 and 50 nanometers, and then we have a polymer shell, peg shell, that makes them colloidally stable. Then we add a functionality, biotin, that binds to streptavidin, I guess most of you know. So it has a very high affinity, and we can measure the K-on rate of that binding under controlled conditions where we should have reaction-limited binding. And what we observe is that, contrary to what you should have if this is purely kinetically limited, the size matters for the K-on rate. If you have the same biotin density from the two particle shells, we should see these curves end up in one line and not in two, depending on size. So obviously this means we don't have the proper limitation. What can this depend on? Well, it can depend on that the particles are actually interacting with the surface that they try to bind to through non-specific forces, like van der Waals and double-layer forces. In order to map out that potential, we need something, a tool, to measure the interaction potential of nanoparticles with the surface, which is not so easy because they scatter very little light. But then, developed by some colleagues of mine, was a very, very flat waveguide, which makes it possible for us to measure scattering from single nanoparticles with such precision that we can look at these gold nanoparticles and look at what they have for distribution close to the surface, and also count for differences, small differences in particle size that influence the scattering a lot. And from that calculate histograms from which we can calculate then the colloidal potential that they have interacting with the surface. And of course measure that this is different for the two different particle sizes, as it should be. Since we have a slight charge on the particles, we introduced that on purpose, we can tune the ionic strength to switch on and off this repulsive contribution in order to then shift what the colloidal potential looks like. So, long story short, if we take this into account now, we know the colloidal potential, and then we normalize basically the probability of groups to be interacting with the surface, and we put that on the x-axis instead. So this is basically the reaction probability normalized, or taking the colloidal potential into account. Actually, all the particles end up on the same curve again. I think nicely demonstrating that the specific interaction is modulated by the non-specific interaction. Yeah, I think I stopped there because I'm running a bit out of time. So this example was not chosen randomly. The colloidal potentials that we engineered for these nanoparticles are about the same that you would expect to have from a virus particle or a 100 nanometer liposome, using simple Hammacher theory to recalculate the van der Waals interactions. So we think it also has biological relevance, not only relevance for people trying to do targeted nanoparticles. So second part of the course now, to be very fast, this is magnetically controlled drug delivery vehicles. So liposomes can be used to deliver drugs. They are nice in that respect because you can put drugs, for example, inside the lumen here and then try to release them out from the liposome at the site of delivery that where you want it to be. This is very nice because most drugs are not well solubilized and they have to be protected from the environment. It's basically the core shell idea again. Problem is that if I try to design a vesicle that is very stable and encapsulate the drugs very well, the release will be quite poor. So the release kinetics are bad. And people have tried to solve that. And one thing that's already on the market since many years is that you use a temperature-sensitive liposome. Basically, you make a lipid composition that has a transition temperature from gel to liquid at a temperature close to body temperature. And therefore, they get leaky because at the phase transition, during the phase transition, you get phase separation from gel to liquid domains. And at the border between those, you have a much higher permeability and drugs can come out. Problem is if you do that, you're leaking drugs all the time because the phase transitions are broad. So it's not a very nice strategy when you have small temperature differences within the body, or you can only induce small temperature difference from the outside. So you're sort of stuck between a rock and a hard place. Very nice encapsulation stability, poor release, or very good release, but poor stability. Of course, nanoparticles can sort of fix this for us, because with a magnetic nanoparticle, I can locally introduce heat by an alternating magnetic field. You are transparent to magnetic fields in its frequency range, and basically flip the spins back and forth. And if I do that, of course, I will have losses, dissipation of heat that is coming only then from the nanoparticle. And now what I can do is that I can put the phase transition temperature of the membrane very high, let's say 55 degrees, as in this example, and then make sure that I have 55 degrees only produced by the nanoparticles, basically melting the membrane here and getting the drugs to release close to the nanoparticle site. Easy enough. Problem is I have to design nanoparticles that can do this. And the nanoparticles then have to be small enough to energetically fit inside the membrane. They have to be very stable shells, not releasing any of the ligands that make them hydrophobic, because then I change the phase behavior of the membrane itself, because they will basically go as small oil or fatty acid chains into the membrane here. So this is something, of course, we could do given that high precision with which we synthesized particles and the anchor chemistry that we developed. In the end, then, one can show such things like which nanoparticles actually fit into lipid membranes, what sizes do they have to have, and this is quickly showing this, that if they're too big, they cluster and they go out and you get micelles instead. If they're small enough, we can put them inside the membrane at controlled concentrations. If we do that, we can then use magnetic fields, like I explained in the hypothesis, that we switch the magnetic field on, we release some drug, we switch it off, there's no drug release, on, off, which makes it possible to arbitrarily release drug wherever and in what amounts that I want, at least in a test tube. Maybe not in the rabbit, we haven't tried, we'll see. The reason why this works is because I'm only heating up and changing the phase behavior of the membrane. I'm not destroying it, which is shown by that the liposomes stay intact with the same size during all of these steps. It's very important, just to emphasize that, to have the nanoparticles inside the lipid membrane. It will be easier for us if we can put them around, but we cannot, because heat transport in water is so efficient and so fast that it's enough to be a few nanometers away from the lipid membrane. I don't have local heating anymore. What happens is I have to heat the entire volume to get the same effect. We can do that too, but that would not be possible in vivo, and it will not be useful for many applications. But if I put the nanoparticles in here, now this is much more optimized since we did this the first time, but I can stay at temperatures for the entire solution which is much lower than the body temperature, meaning, well, if it's in body temperature, that's the baseline, but I don't heat the solution above temperatures which would be dangerous in a biological environment. So it really requires this type of engineering. In the end, we looked at this as well, all the factors that influence this and what we can control and what we cannot control. So actually the ones that we built for trying applications in the lab, they are optimized in such a way that we have only a few nanoparticles per liposome. But we make sure, since they are statistically distributed, that we have at least one per liposome optimally. And we can therefore tune the release rate with the number of particles, with the pulsing sequences that we use for magnetic fields. We can tune it by the lipid composition, where we put the phase transition temperature and so on. We have a lot of handles to play with this. And the nice thing is that we can also incorporate them into lipid systems, which are the same formulations with PEG lipids and cholesterol and so on that are used already in drug delivery vehicles. And the stability can also be such that we can store them for weeks with the drug inside, even when we've incorporated nanoparticles for triggered release. So to summarize up, I'm now at 25 minutes. What I hope to have conveyed to you is that it is possible to very carefully design core shell nanoparticles to the extent that we control the colloidal interactions and therefore can focus on the nanoscale properties that these can give to us in experimental situations, be them quantum dots, plasmonic, or magnetic, as I showed you today. If we do that, it's also possible then to vary the functionality that we have on the shell, and also, which I didn't show you too much, to switch these colloidal potentials as well. Decide when they're non-interacting, when they're interacting, when they present a biological function, and when they do not, by, again, controlling the type of polymer shells that we're using. If we build the particles carefully enough, we can also use them as supramolecular than building components. This is actually more difficult, I would say, and build them into liposomes, for example. What we've done, and I didn't have time to show you today, is that we actually go beyond liposomes and we make vesicles that have block copolymer and lipid membrane windows in them, which affords us much higher stability, but we can still switch, for example, permeability and so on. This is the, sort of the next step in evolution that you can do with this. And with that, I would just like to thank the people that actually did this, because it was obviously not me. So these are students that were involved in everything from the synthesis, developing the early synthesis, to the more colloidal aspects of the whole thing, and of course the postdocs that were really instrumental for doing this. I would especially like to point out Ronald, who's been driving since the nanoparticles, and Anders, who has been really driving behind both the very bad poster I presented yesterday in microbial interactions and these ideas about multivalency in biological systems. And then, of course, the collaborators. Eddie here was very instrumental for the cyclic polymers, and we're still working together on topological polymers, on nanoparticles, and Fredrik, who contributed the waveguide technology that we used, and Thomas Helbich, which is the medical professor that we're collaborating with, and of course all the funding agencies for giving me at least enough funding to survive, and I hope they give me more. Thank you for your attention, and I hope for your questions.
Hello, please. You said that these nanoparticles can stay in the organism for weeks. What is the biodegradability of such nanoparticles?
There was a slight misunderstanding if I said that. So they are stable inside the liposomes for weeks without releasing drugs. If we put these particles in, they are much more, if we engineer them to have no interactions, no protein functionality at all, then they have a much longer circulation time than if they don't. If we put proteins on, they're actually stable in circulation for not much more than 30 minutes. If we don't put that on, there's a long time since we did these experiments, so they're not really built on the best particles that we've made, but then it's in, let's say, 12 hours or so in a rabbit. The biodegradability is quite nice, so since we can choose the polymers that we use to have very low toxicity, and this is the most toxic part we have. And the amount of iron oxide that you can put into the body is really high. This is in milligrams per kilo without breaking any guidelines or having any bad effect, probably much more than that. We only do cytotoxicity studies, and usually they are like fertilizers for cells, rather than we never show a cytotoxic effect if we didn't use a cytotoxic polymer on the nanoparticle. Don't know if that halfway answers your question.
Okay.
So, very nice talk. Congratulations. I want to ask about your polymer functionalized magnetic particles. So, in an ensemble, basically you have magnetic particles embedded in a meshwork.
Yeah.
The question is, could you in principle use this to measure local tissue stiffness somehow by measuring the coupling in between the particles with MRI, for example?
Not with the particles that I showed you today, because they're in the superparamagnetic range, which means I need to have magnetic field strengths which are higher than the 9 Tesla magnets even that you use for clinical MRI, or high-end clinical MRI today. Of course we can, and we have done that too, but not as much study. We can also make particles bigger, make them at least 25 to 30 nanometers. And since the magnetic moment scales basically exponentially with the volume, these are much more magnetic than the ones that we have here. Then you can start to pull and do such experiments with them as well. Of course, if you, with MRI as well, you don't really have the resolution, but of course if you have a clever way of imaging positions of these magnetic nanoparticles, you can do fluctuation measurements of stiffness locally as well. And then maybe these particles would work as well as just reporters. But if you need to also create a stimulation through the particles, you need to design the particles bigger, but it would be the same kind of concept.
very much for introducing me and for giving me opportunity to be here and presenting you a piece of work that we have done together with our Polish colleagues from University of Łódź. And as you can see from my title, so I will stay within this topic of nanoparticle-mediated drug delivery. But I will speak about another type of nanoparticles, viewing them as a vaccination platform. Okay, and we were focusing to the interaction of those nanoparticles with biological membranes. Those nanoparticles, they were synthesized by a Spanish chemist. There's a plenty room. Yeah, I'm too small. Okay, thank you. So there's a plenty of room at the bottom. That was the title of Richard Feynman's presentation. Lecture he had given in 1959 at American Physical Society meeting, and he was actually one of the first who pointed to the uniqueness and enormous potential of studies in nanometric scale. So creating nanoparticles at this scale, they will possess many unique properties, and they will get into the size of biological molecules such as protein and nucleic acids. So main idea of using nanoparticles for various biomedical applications is to better control biological processes, improve disease detection, therapy, or prevention. And there are different types of nanoparticles currently being synthesized, either of organic or inorganic nature. So there are liposome, polymer, dendrimer, or those already mentioned core shell inorganic particles. And they all offer fundamental solution to fundamental problems in a drug industry that are connected with low solubility of drug as well as lack of their targeting. So drug can be encapsulated either inside of nanoparticles or on the surface, and this surface can be modified also by other molecules to improve their detection or targeting. And I will focus to dendrimers. So this is special class of hyperbranch polymer structures. This word dendrimer, it comes from two Greek words, dendron, which means tree, and meros, which means part. So this structure really looks like tree, and this architecture consists of three main parts. So there is core, those branching units that are made by iterative sequences of reaction steps, and in which each reaction results in a so-called new generation. And a final generation, so there are terminal groups. Drug can be encapsulated either inside those cavities or on the surface. And this surface can be modified, as I mentioned before, by various other molecules to improve delivery and support the therapeutic activity of transported cargo. And we were working with these two types of dendrimers. These are carboxylene dendrimers. They're cationic molecules, and originally they were synthesized for complexation and delivery of sRNA and oligonucleotides. And we were working with these two types. So this is carboxylene dendrimer that has carbon-silicon bonds. This has oxygen-silicon bonds. And we were trying to complex them with HIV-derived peptides. HIV, as we know, is a human immunodeficiency virus that is responsible for acquired immunodeficiency syndrome. This is also known as AIDS. And this virus attacks our immune system, our immune cells, use these cells for its replication and destroy them. And just looking at the current situation, so there is approximately 36.7 million people currently living with this virus. And some statistics numbers. So number of newly infected people with this virus, like a few years ago, and up to now, there is decreasing trend, which is good, but still like last year, there were 1.8 million people being newly infected, and approximately 1 people died in 2017 because of AIDS-related diseases. So it means that HIV/AIDS remains still a serious problem worldwide. So let's have a closer look to this virus. So it's a structure approximately 100 nanometer in diameter, and it consists of 2 single-stranded RNA that are enclosed together with other enzymes that are needed for its for formation of new virion into this nucleocapsid. And all these components are enclosed by lipid bilayer from host cells. And on the surface, we can see glycoproteins such as GP120 and GP41 that are responsible for binding to the target cells and for entry. Target cells, so this virus attacks mostly T cells, macrophage, and dendritic cells. So when this virus gets close to the surface of these target cells, it fuses with the membrane of cells, then all the components are released. RNA molecule is transcribed into the DNA, which is then integrated into the DNA of host cells. And then RNA is formed as a genomic RNA, and that makes a new protein. All these components are transferred towards the plasma membrane where new virion is formed. And I'm mentioning this viral cycle because currently HIV infection can be treated by antiretroviral agents that act on different stages of this HIV life cycle. So there are, for example, entry inhibitors or nuclear reverse transcription inhibitors, and they can really decrease viral load, but there are still limitations that are connected with this medication. These agents, they cannot really remove all virus from our body, so they cannot really provide successful therapy as well as prevention. So there is a question if there is some vaccine that would protect us against this infection. And currently there is no vaccine to prevent HIV. So we were focusing to this topic. And in general, there were many attempts to develop some efficient vaccine compounds. And these can be done by using either live inactivated microorganism or taking some part of this pathogen just to create immune memory. And I will focus to peptide-based subunit vaccination strategy that is targeted to dendritic cells. Dendritic cells are special immune cells, and their role is to capture peptide and process it on the surface where then is expressed by this major histocompatibility complex. So T lymphocytes can detect these peptides, and then humoral or cellular immune response can be generated. But it's not so easy. Most difficult is this part because peptides just by themselves they are not easily transferred to the cell membrane. Those peptides, they are generally safe and able to induce very specific but weak immune response. So they need some immunostimulants, also called adjuvants. So those dendritic cells represent one of those adjuvants, but we need another help to sufficiently deliver these peptides across the cell membrane, and nanoparticles can serve as carriers for these peptides, so they can deliver and support biological activity of those transported material. And we were using for this purpose those dendrimers, and we were focusing to their interaction with biological membranes. But before studying these interactions, so we had to check if those dendrimers are able to complex those peptides. And this characterization is was made and is basis of this paper. So here we checked those dendrimers, complex them with different types of peptides and characterized them. So we selected some of them. And then we will now focus to their interaction either of peptides itself or dendrimers or those complexes with biological membranes. Because as we know, so biological membranes plays an important role in a cell life. It's a barrier that is selective. It controls movement of substances that goes out and into the cells, but it's really complex structure. And to analyze interaction of our dendriplexes, we were using lipids, model lipid membranes. So we analyzed this interaction on a molecular level using either lipid monolayers or bilayers. And this is the summary of material that we used. So we were working with those two types of dendrimers, 3 types of HIV-derived peptides. And for making those model membranes, we were using this kind of lipids, zwitterionic, negatively charged, as well as pegylated. And the methodology procedure, so we were creating monolayers using Langmuir-Budget through. So we're creating monolayers by spreading lipids on a air-water surface at a certain surface pressure, which was approximately 30 millinewtons per meter. That's the physiological value of normal cells. And then through a small aperture inside the measuring cell, we injected our compounds, and we were checking changes in that surface pressure. And in the case of lipid vesicles, so we were analyzing fluidity of this membrane by measuring fluorescent anisotropy, and we were monitoring region of polar head groups as well as hydrophobic core using 2 commonly used fluorescent probe, TMA-DPH, that is located in hydrophilic region, and DPH that is located inside the hydrophobic core of membrane. And this kind of model membrane, this liposome, we were also analyzing by means of size and zeta potential measurements. And now I will move to the results. So firstly, thinking about those dendrimers as carriers of some material, we should check if those dendrimers may interact just by them itself with biological membranes. And here are the results from fluorescent anisotropy measurements using different types of lipid vesicles, using different types of lipids. And this parameter, when it increases, it means that membrane fluidity is decreasing. So membrane is getting more rigid, and we can see that steady-state fluorescent anisotropy increased for all cases, suggesting that those dendriplexes interacted with polar head groups region as well as with hydrophobic core of those membranes. And interaction was much more stronger in the case of negatively charged lipid. And what was interesting was also different behavior of these two dendrimers, because in the case of carboxylene dendrimers composed of oxygen silicon bonds, so interaction was much more stronger in the hydrophilic region, and dendrimer with carbon silicon bonds interacted much more stronger with the acyl change of lipids. So looking now at the results from monolayer study, they corresponded quite well with those when we compare them with anisotropy results in hydrophilic region. So interaction was again stronger in the case of negatively charged lipids, and the changes were much more pronounced in the case of this type of dendrimer consisting of oxygen silicon bonds. So result of this study are that yes, dendrimers could interact with membranes, and they have ability to complex HIV-derived peptides. So let's check them now. Let's check those dendriplexes, how they can interact with hydrophilic part of lipid vesicles. And what was interesting that when we increased molar content of PEG, the interaction was weaker. And in the case of hydrophobic core, these particles didn't interact with liposomes composed of that PEG lipids. Slight increase we observed for just pure DMPC liposomes. And those results were also supported by results and measurement performed on lipid monolayers. So increasing the molar amount of PEG didn't cause such a higher changes of surface pressure comparing to the pure DMPC monolayer. And those monolayers were also visualized by Brewster angle microscopy. I don't know how good visible it is, but here, this is monolayer where this is pure DMPC monolayer, and this is after addition of dendroplexis. So this DMPC monolayer is in a liquid content state, and we can see some white spots here that correspond to lipid microdomains. And in the case of application of our dendroplexis, we can see region of disturbed monolayer surface. Pegylated monolayers, they were more homogeneous, and after addition of dendroplexis, there were also observed some kind of domains of different size. So taking all together this story and summarize it, so HIV remains a serious problem worldwide, and research in the field of HIV vaccine still continues. We were focusing to the peptide-based synthetic vaccine, and those peptides, we were trying to support their immune response by using nanoparticles as carriers. We were testing carboxyl and dendrimers and testing their interaction with biological membranes. And as we could see, HIV-derived peptide itself, they didn't interact with those biomimetic membranes. Carboxyl and dendrimers interacted with those membranes and were able to complex peptides. So those complexes interacted as well with those studied membrane models, and interaction was much more stronger in the case of negatively charged membranes. And in the case of pegylated membranes, dendroplexis interacted mostly with the hydrophilic part, with the ability to absorb on the lipid monolayer with this order. So analyzing this interaction, it means that those complexes interact with biological membranes mainly due to electrostatic forces, and are also dependent on the ordering of lipid films. And obtained data can be useful for optimization and potential of these carboxyl and dendrimer as carriers for HIV peptides. And actually, I would also like to mention another type of particles. So we are spreading this topic, and this is also kind of like core shell particle. So those chemists I mentioned, they are synthesizing those dendritic nanoparticles, so they incorporated gold core, and on the surface there are those carboxyl and dendros. So now we are testing and studying biophysical characterization of these complexes with, again, those HIV-derived peptides. But we are currently working on this. So I would like to acknowledge and thank you agencies for financial support, and also all to my colleagues.